So today we look at the chromosome structure. So when we have to start about the discussion about the chromosomes, so first question comes in our mind that where these chromosomes are located. So as you know, when we talk about the chromosomes, so we say that the chromosomes are located in the nucleus of a cell. But how these chromosomes are being made? What is the chemical constitution of this chromosome? So that is the important part. So whatever the entire thing present in the nucleus, we call that thing as a nuclein, or we can also call that as a chromatin. So what is this chromatin? Okay, chromosomes are generally, it's a chromatin thing. So what is this chromatin? So chromatin is a nucleoprotein complex. In a nucleus, the chromosomes are present in the form of chromatin. Okay, it is one of, that means the fine structure of that chromatin, we call it as a chromosome. So this chromatin means it is a nucleoprotein complex. It's a constituent of DNA and protein. It's a constituent of DNA and protein. So generally I can tell you that the chromosomes are made up of not only DNA, but they are associated with the various proteins. They are associated with the proteins, and this entire structure we call it as a chromatin. So the chromosomes, as I said, they are associated with the protein. So whatever the proteins are there associated with the chromosomes, which is made up of DNA, that is deoxyribonucleic acid. So whatever the protein are associated with that DNA, so it is present in 2:1 ratio. So if there is a so twice the amount of protein is there in a chromosome. So if we consider there is a 1 unit of DNA in a chromosome, so twice the amount of protein is there. Okay, and out of this total protein, the histone protein is half of that. Means if you see the ratio of histone proteins to the DNA, it is 1:1. So what are these histone proteins? These histone proteins means the proteins which are associated with the DNA. As you know, the DNA is negatively charged because of that phosphate group. Whereas these histone proteins, actually it is rich in basic amino acids. The histone proteins, they are rich in basic amino acids. So what are these basic amino acids? These are arginine, histidine, and lysine. And they are positively charged amino acid. They are positively charged amino acid. So in general, histones have a more positive charge. And in contrast to that, DNA is more negatively charged due to the phosphate group. That's why there is a tight interaction between histone proteins and DNA in a chromatin. That's why there is an association of histone protein along with DNA. And this present in the ratio 1:1. Okay. So chromatin. So we are talking about the chromatin. So there are several levels of condensation. As I told you, the chromosome is the very fine structure of that chromatin. So there are various levels of condensation of that chromatin. So 10 nanometer nucleosome like beds on string model is there. So we call it as a beds on string model, that is of 10 nanometer. Okay. So structural unit of this chromatin is called as nucleosomes. Okay, they are the primary structural unit. Nucleosomes are primary structural unit. I will tell you about what are the nucleosomes. Okay, so the nucleosomes are the primary structural units of that chromatin. Then 30 nanometer fiber. Okay, that is a nucleosome interaction. So nucleosome is the primary fundamental structure of the chromatin, whereas 30 nanometer fiber is secondary level of compaction. Okay, secondary level structure. So you can see there. So at left side, you can see the DNA. You can see the presence of DNA inside the nucleus. Okay, presence of DNA inside the nucleus. So how this DNA, how these chromosomes are made up of DNA? You can see the different level of organization of DNA. So first there is 30 nanometer fiber. So in 30 nanometer fiber structure, you can see there is one circle. Okay, there is one circle there, yellow color circle. So that is a nucleosome. That is one nucleosome. So that is 30 nanometer fiber. Okay, 30 nanometer fiber. Then we can see weights on string model. That is 10 nanometer fiber. That is 10 nanometer fiber. So nucleosomes. Okay, I told you the basic unit of that chromatin is nucleosomes. And then thereafter, you can see, and if you look at the one nucleosomes, you can see the double stranded DNA or DNA is wound around that nucleosomes. Okay, so DNA, the nucleosomes are surrounded by this DNA. Okay, nucleosomes are surrounded by this DNA. So this is the fine structure where we look at the DNA. Okay, so first is what are the different level of structural organization of this chromatin. First is 30 nanometer fiber. Then there is a 10 nanometer fiber. 10 nanometer means weights on string model, and nucleosome is a basic unit of that chromatin. So we talk about the nucleosome. We say that it is a basic unit of an chromatin. Okay. So here you can look, if you look at this thing, in the first slide only, I have explained that the ratio of histone protein to the DNA is 1:1. So in this slide, you can see that there are different histone proteins like H2A, H2B, H3 and H4. Okay. So each of these histone protein, H2A is one of the histone protein, H2B is another histone protein, H3 is other histone protein, and H4 is the other histone protein. So H2A, it is present in the two copies. H2B, it is also present in the two copies. H3, H3 is also present in two copies. H4 is also present in a two copies. That's why we call it as an octamer. There are 8 molecules of proteins. So each histone protein is represented twice. That's why we call it as a core of 8 histone. So a nucleosome, which was the basic unit of chromatin, it contains 8 histone proteins. So 2 molecules of each histone, H2A, H2B, H3, H4. So whatever we are talking about, it is for one nucleosome. Okay. So nucleosome contains 147 base pair of genomic DNA wrapped around a octamer of histone proteins. So one nucleosome is wrapped with 147 base pair of genomic DNA. Okay. So this is nucleosome. As DNA is associated with the histone proteins, as I told you. So this is the nucleosome. Here we can see the structure of nucleosomes. So two nucleosomes are together joined by the linker histones, and that histone we can call it as a H1 histone. Okay, H1 histone, which is present on the linker DNA. So two nucleosomes, we know there is a model. It is a based on string models. So nucleosomes, there are multiple nucleosomes. Okay, so these two nucleosomes are joined together by other histone protein that we call it as a H1 histone protein. Okay, H1 histone protein. And in a nucleus, there is a presence of one nucleosome, 147 base pair of DNA. And when we talk about this chromatin, which is the DNA protein complex inside the nucleus, so this chromatin has been divided into the two parts, that is heterochromatin and euchromatin. So heterochromatin means it is the genomic region, it is the DNA which is transcriptionally silent. Means if you see, I will give one good example. If you know about the human genome, okay, if you have heard about the human genome sequencing project, actually it has been completed in the year 2002. Okay, human genome sequencing project has been completed in the year 2002. So when human genome sequencing project has been completed, then we came to know that there are 23,000 genes in a human being. Okay, in a cell, in a nucleus, there are 23,000 gene on that means on in an entire genome. But there there was again one interesting thing has been observed, that is 97% of human genome is non-coding. What it is? Non-coding means it is transcriptionally silent. It is heterochromatin. It is not code for any type of protein. Only 3% of genomic DNA it is active, means it express, means it forms an mRNA, it forms a protein. Only 3% of total genome or total DNA is the active DNA, or we can say that it is expressed part of a genome, or in other way we can say that it is a euchromatin, that is transcriptionally active. So in human beings, if you see 97% of genomic DNA, it is transcriptionally silent. It is the present as a heterochromatin. Okay. And only 3% is of euchromatin, which is transcriptionally active DNA. Now I am talking about what euchromatin and heterochromatin. So in euchromatin, I said that it is a transcriptionally active region, means it is a gene rich region. So whatever 23,000 genes are there, that constitutes only 3% of total genome. Okay, 3% of total genome, and it has a unique DNA sequence. Whereas heterochromatin, if you look at the heterochromatin, it is gene poor region. There you can't find any genes. It is a less transcriptionally active, where the repetitive DNA sequences are there. And this heterochromatin occupies 97% of the DNA or 97% of the genome in case of human beings. So we have seen about the chromatin. We have seen about the how the structural organization of this chromatin, where the DNA is associated with the various proteins, and we call them as an histone proteins, which are present in 1:1 ratio. So what are these histone proteins? It is H2A, H2B, H3, H4. So a nucleosome contains 8 core histone proteins. Okay, 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3, and 2 molecules of H4. I also told you that these histone proteins are basic in nature. They are positively charged. They are having higher positively charged because they are rich in basic amino acids like arginine, histidine, lysine. Whereas in contrast to that, DNA is negatively charged, and that's why there is a tight interaction between these histone proteins and DNA, and we call that entire structure as a chromatin. Okay. So now if we look at the chromosomes, so chromosomes are the rod-shaped filamentous body present in the nucleus, which become visible during cell division. Okay, which become visible during cell division. So only when there is a cell division, then you can find out the number of chromosomes, then you can study the shape of the chromosome, then we can study the number of chromosomes. So they are the carriers of gene or unit of heredity. You know, Sutton and Bowry, we have proposed the chromosomal theory of inheritance. Means the chromosomes, or in other words, we can say the DNA. It is the unit of heredity. Okay. So the chromosomes, where the genes are located, means these chromosomes are made up of DNA, and genes are located on that. Means it is a particular segment of a DNA. So genes are located on the chromosomes. So the chromosomes were first described by Strasburger in 1875. You should remember the name of scientist. And the term chromosome given by Valdier. Okay. The term chromosome is given by the scientist Valdier in 1888. Whereas the chromosome were first described by the scientist Strasburger in 1875. So the chromosome, chroma means color and soma means body. Okay, it's color body. That time it was named as chromosome because they have seen it as a color body. So in the first circle, there we can see one chromosome. Okay, so this is before mitosis. This is before mitosis, and you can see one chromatid per chromosome. So one chromatid per chromosome, and you can also see the centromere is there. Okay, so why, see, before mitosis, if you look at that chromosome, that first image, first circle, so the chromosome which is represented, the chromosome structure which is before mitosis, you know, mitosis is one of the cell division which is occur in the somatic cells. The outcome or significance of mitosis is the development of or production of a two genetically identical daughter cells from a parent cells. Okay, so the mitosis is very important for the growth and development. And whenever there is a formation of the two daughter cells, two genetically identical daughter cells, we call the mitosis process. When the mitosis process, outcome of the mitosis process is the formation of two cells which are genetically identical. Which are genetically identical. Whatever the product of meiosis is there, we get an haploid cells, haploid gametes, which are genetically dissimilar. But in case of mitosis, whatever the cells or whatever the cells are being formed, new cells are being formed from the parent cells, they are genetically identical. That is the outcome of mitosis. Okay, so when we say that they are the genetically identical, means the whatever the genetic content of that parent cell will be there, that has to be equally distributed to the two daughter cells. What I am saying, it should be equally distributed to the two daughter cells. Then only we call that daughter cells as a genetically identical. You know that during the growth and development, or whenever there is an wound or injury to the cells, so immediately it will get repaired. It is by the mechanism of mitosis. And if you know, if you so if you see that in a day, around 10 raised to 11 million cells are died in a day. So that many cells are getting synthesized new in a day. So by the process of mitosis. So from parent cell, if there should be an equal distribution of the genetic material to the daughter cells. So the parent cells should synthesize the more genetic material. Okay, it has to replicate that genetic material. It has to duplicate that genetic material. The genetic material should be twice in the amount. If it has to be get distributed evenly in a daughter cell. I will give you one example. I'm talking about the genetic constitution, but what about the cytoplasmic content? Suppose a parent cell, if it carries the 10 mitochondria, 10 chloroplast, and you know mitosis. So after the mitosis, there is a formation of the two daughter cells. So if that 10 mitochondria, that should be evenly distributed to the daughter cells. Means it's not like an 10 mitochondria is getting distributed as like 5-5. It is getting distributed as a 10-10. How it happens is because of cell cycle. If you look at the cell cycle, there is an interphase. During that interphase, there is a G1 phase. You know, interphase is constituted G1 phase, S phase, G2 phase. G1 phase where the organelles are getting synthesized because it has to get distributed to the daughter cells. Then during the S phase, DNA is getting synthesized because that parent cell has to distribute the genetic constitution, DNA or chromosomes equally to the daughter cells. That's why there is an S phase, that is a DNA synthesis phase. Okay. So if you look at the middle circle, you can see there a chromosome, one chromosome, that is after replication. That is after replication. You know, DNA synthesis can happen only in S phase of cell cycle or S phase of interphase. So there DNA synthesis can take place. So what is this DNA synthesis means? The sister chromatids is getting synthesized. So you can see there how the chromosomes is look like. So there are two chromatids, and these two other chromatid we call it as a sister chromatid. What it is? sister chromatids, because this two chromatid has to get separated individually during the anaphase to the different poles, and that's why the daughter cells will carry the equal amount of genetic material. So once it will get separate, that two different chromatids which has been synthesized during the S phase, that means DNA has been synthesized, chromosome has been duplicated. So after the S phase means there is an G2 phase, and then it is a mitotic phase, that is prophase, metaphase, anaphase, telophase, and then it will be followed by cytokinesis. So during this anaphase, you know that two sister chromatids move to the opposite poles. Okay, two sister chromatids. How the sister chromatids are being formed? That is because of DNA synthesis. That is because of DNA replication. So the two chromatids move towards the opposite poles. And after telophase, you know, cytokinesis happens. Means one cell carries the one chromatid, another cell will carries the other chromatid, and we call that chromatid as a chromosome at that time. We call that chromatid as a chromosome at that time. So in middle U structure that you can find this is during the S phase, after the S phase, that is the DNA synthesis. Okay. So as I told you that the chromatids, means sister chromatids, it gets synthesized during the S phase. Those sister chromatids and that other chromatid are joined together at the one center, and then that center we call it as a centromere. We call that center as a centromere. Okay. So two sister chromatids are joined together, sister chromatids joined together at one point, and that point we call it as a centromere. And we know that at the centromere, so during that anaphase, so that two different chromatids are separated together separated at opposite poles. Okay, the same thing whatever I explained. So this is the structure of chromosome. If you look at the chromosome, so this is after S phase, okay, DNA synthesis, and this here we can see the sister chromatids, that is the duplicated chromosomes, because it has to get separated, it has to get divided to the two daughter cells equally. Okay, so that is after S phase, DNA synthesis phase. So we can see that histones are there, DNA is there. So these chromosomes are generally constitute of a DNA as well as histone proteins. Now, so we have seen about the chromosomes structure. In general structure, we have seen about the chromosomes. So you know that there are various chromosomes in a cell. So if you say the human beings, the total number of chromosomes are 46 in a human being. Sorry, I'm sorry. Actually, it has been moved. Just a minute. So what I was talking, the total number of chromosomes in a human beings are 46. Means we say that 2n is equal to 46. Okay. And out of this 46, there are 22 pairs of autosomes and one pair of sex chromosomes like XX and XY. So 22 pairs of autosomes means we call that 22 pairs. Pairs means each chromosome has its homologous chromosomes. And from where that homologous chromosome is coming, it is coming from, one is coming from paternal side and one is coming from maternal side. Here we can see 1, 2, 3, 4, 5, 6, 7. So all 22 chromosomes are represented. Okay. So here we can see the one number chromosome. So it has its homologous chromosome. Second chromosome. This is a pair of chromosome. Okay. So homologous chromosomes are there like 2, 2 dash, 3, 3 dash, 4, 4 dash, 5, 5 dash. Means this homologous chromosome means one chromosome is coming from one male gamete, another chromosome is coming from female gamete, and then there is a union that's why it formed 2n, that is diploid. Okay, diploid organisms. So particular chromosome complement of an individual or related group of an individual is defined by the chromosome size, morphology, and number is known as karyotype. So what is the karyotype? Karyotype means it gives us the size of chromosomes, karyotyping, Okay, we can find out the size of the chromosomes. We can find out the position of centromere. Okay, size, shape, position of the centromere. So all these things can be find out in the karyotype. We call it as a karyotyping. You can see the shape of the different chromosomes there. This is with respect to human being. This is with respect to whatever the karyotype has been given to you. That is the study of shape, size, centromere position of a chromosome. So you can see the shape of the various chromosomes are that is different, but of homologous chromosome is almost similar. Okay, so that is the karyotype. So actually this karyotype is carry-on. It is derived from a Greek word carry-on, which means nucleus. Okay, carry-on means nucleus. So karyotype is represented as ideogram. So when the haploid set of chromosome of an organism are ordered in a series of decreasing size, it is said to be a ideogram. So the diagrammatic representation of karyotype, we call it as a ideogram. Okay, diagrammatic representation of the chromosome. So whatever we have seen, the diagrammatic representation. So this is the diagrammatic representation of the various chromosomes. So we call it as a ideotype. Why? What we call it as ideogram. Okay, ideogram. Okay. Sorry, how it is moving? Just a minute. So in a chromosome, you know that all the chromosomes are differ in their structure, they are differ in their size, and of course they are having the different genetic constitution. Okay, that means if you see the chromosome gene mapping or genetic mapping, so you should know that which genes is located on which chromosome. Okay, suppose there is a XY. So when we say that it is karyotype, so ideogram means it is a schematic representation of that chromosome. Okay. Then Levitsky, who is the first person to define the karyotype as phenotypic appearance of the somatic chromosomes. Levitsky. Okay, in 1931. So here we can see the different forms of a chromosomes. I told you that karyotype means where you can study the shape, where you can study the structure, and where you can study the location of centromere. So centromere means the position or the center where the chromatids are being attached. Okay, so here we can see some chromosomes are telocentric. Okay, some chromosomes are acrocentric, means centromere is located at some one end. In telocentric, centromere is located at end. In acrocentric, centromere is located near the end of chromosome. Centromere is located near the end of chromosome. In telocentric, centromere is located at the end of chromosome. In acrocentric, centromere is located near the end of chromosomes. Near the end of chromosomes. And in submetacentric, so in metacentric chromosome at the right side, you can see D. Okay, metacentric chromosome. So centromere is exactly at the middle position. Okay, and you know there are two arms of a chromosome like P arm and Q arm. Okay, P arm means short arm, Q arm means long arm. Okay, so telocentric means where it is a type of chromosome. It is a that means structure you can say whether it is a telocentric chromosome, acrocentric, submetacentric, or metacentric. That we can classify based on this position of centromere. If centromere is located at the end of the chromosomes, then we call it as a telocentric. If centromere is located near the end of chromosomes, then we can call it as a acrocentric. If centromere is located near the middle position of a chromosome, near the middle position of chromosomes, then we can call it as a submetacentric. And exactly at the middle position, if centromere is located exactly at the middle position of a chromosome, then we can call it as a metacentric chromosome. Okay, and short arm of the chromosome is denoted by small p letter, whereas long arm of a chromosome is denoted by small q letter. So generally, whenever we study, so you know that the rice is having different chromosome numbers, wheat is having different chromosome number, cotton is having different chromosome number. So all plants are vary their chromosome number. Similarly, the animals are also having the different chromosome numbers. So generally, we take the root samples, we take the root tissues, we stain with some chromosome stains. We use this different stains to stain that chromosomes, and we can find out the that is actually stains for the chromosomes. Okay, and then we can see the metaphase stage. Okay, and then we count the chromosomes, how many chromosomes are there, how they looks, that means what is the shape of the chromosome, what is the centromere position. So this is the karyotyping. Okay, this process we call it as a karyotyping. If I will that slide actually, so whole this process is called as karyotyping. And the representation of the different chromosomes, the representation of the various chromosomes, diagrammatic representation of various chromosomes, that we call it as a ideogram. That things we can call it as a ideogram. Means if rice is having uh, if rice is having 42 chromosomes, if rice is having 42 chromosomes, means all 42 chromosomes are represented in one such a diagrammatic things. Can you see here? So they are arranged based on their size, based on their shape. So these things we can call it as a ideogram. Okay, so longest in a karyotype, longest chromosome, it is on the extreme left. So here we can see the longest chromosome is on extreme left, that is one number chromosome, whereas shortest chromosome you can see at the last. So shortest chromosome on extreme right, sixth chromosome at the extreme right. So that is the arrangement of chromosomes, and that is the study of that study we can call it as a karyotyping. Okay, and ideogram means it is an actual diagrammatic representation of that chromosomes. So you can look whenever, now you can look at the chromosome. So you can see some are the darkly stained region, some are the light stained region on a chromosome. So darkly stained region means they are the heterochromatin, whereas light stained region means euchromatin. As I told you, euchromatin is transcriptionally active region of a chromosome or DNA, where it is a gene rich region. Euchromatin, it is transcriptionally active region of a genome, whereas heterochromatin, it is transcriptionally inactive, where you can't find many genes. It is the gene poor region. I have given the example of human genome, where 97% of genome is heterochromatin, means it is non-coding, means it is transcriptionally silent. Only 3% genome is code for particular protein or particular RNA. So this is ideogram. So what are the advantages of karyotyping? Here we can revise the structural features of each chromosome, help in studying the chromosome banding pattern, helps in identification of chromosomal aberrations, that we'll see in the next slides. What are these chromosomal aberrations? Then we can also diagnosis of prenatal genetic defects. We can also study the various genetic disorders, various genetic diseases, and we can also study the evolutionary changes. So this can be happen because of this karyotyping. So we have seen that the short arm of the chromosome is denoted by small letter p, whereas the long arm is denoted by the letter q, small q. And these short arm and long arm are joined together at the position we call it as a centromere. And we have also seen the classification of chromosome based on the position of the centromere, like telocentric, acrocentric, submetacentric, or metacentric. So you can see the bands there. As I told you, there are darkly stained region, light stained region in a chromosome. So darkly stained region, we call that as an heterochromatin. Light stained region, we can call it as a euchromatin, that is gene rich region or active region. So what are these bands on a chromosomes? Okay, the bands on chromosome. So we can see that there are different bands on chromosome, like you can see the color differentiation there. So in 1958, Kasperson, he has first published the paper describing the use of pinocrine mustard to stain the chromosomes. So Casperson was the first person who stained the chromosome. Which dye he has used? Quinacrine mustard. Quinacrine mustard. Then Paris report in 1971 was the first attempt to provide nomenclature for chromosome banding in any species. So what actually you have seen there are various bands on a chromosomes. We have seen one ideogram of human beings. So what these bands, what idea gives us, what idea that can come from that bands. So we'll come to know that occurrence of repetitive DNA. So repetitive DNA you can find in heterochromatin region, which is dark stain region. Differences in base composition of DNA that can be found out with this various banding technique. Difference in the protein component that can be found from this banding technique, and difference in the degree of packing of DNA complex. Okay, so this can be studied by using this various banding technique. So what are these chromosome banding? The artificial production of such a bands by treatment with specific dyes like quinacrine mustard. Okay, with specific dye is referred to as chromosome banding. You know, chromosomes are having whenever we talk about the chromosome, so first they will be differ in their protein constitution. Second, they will be differ in their ATGC, means purine and pyrimidine content. Third, the chromosome region may be heterochromatin or it may be euchromatin, that is transcriptionally active region or transcriptionally inactive region. So all these things can be studied by the chromosome banding. So these are the various techniques used to study the chromosome structure or chromosome, presence of heterochromatin, presence of euchromatin, presence of repetitive DNA in a chromosome. So these are the various techniques, and all this technique uses the various dyes like G bending, GMSA dye is used, Q bending, quinacrine dye is used. There is one other technique that is reverse bending. C bending means centromere, where centromeric position is getting stained. You know, the centromere where that chromatid binds. Okay, so centromere, C bending means where the centromeric region is getting bind. Then there is an N bending, that is a nuclear organization region is getting stained by using various dye. So as I told you that chromosome is generally, if you see the chromosome structure, there is a short arm, there is a long arm. So short arm generally is called, denoted by the small letter P, and P stands for PT. Whereas long arm is denoted by the small letter Q. Okay, and that is separated by a centromere. So here we can see the short arm, that is P arm, long arm, that is Q arm. Okay, so short arm. So here we can see so how the nomenclature is given. So banding position like 1, 2, 3, 4, 5, and short arm is denoted by the small letter P, whereas long arm is denoted by the small letter Q. And this various position of the banding or bands on a chromosome, they can be measured by the nomenclature like bands P11, band P23. Okay, so the names are given to the various bands like P11, P12, Q11, Q12, like that. And you can see the centromeric position there. So here we can look at the how the cytogenic map. If I had to say that, okay, XYZ, X gene is present on the chromosome number 5. Okay, but at which position it is located? So like 8q31.2. So the how the location is given, how the position of chromosome is getting indicated by this on the cytogenetic map. So it is like 8q31.2, means this indicate that gene is on chromosome number 8. So how, see, cytogenetic map of gene is, here we can see the example how the gene is located 8q31.2. So where 8 denotes the chromosome, on which chromosome the gene is 8. Q means, you know the what it is, long arm, Q arm of the chromosome. Then 3, band number 3, sub band number 1, sub band number 2. Okay, there you can find that gene. So that is the cytogenic genetic map, or that is the location of a gene. Okay, that can be denoted by like this. You can see that 8q31.2 means this gene is located on the chromosome 8 on the long arm, third band, and first subband and subband number 2. So as I told you, there are various banding techniques. So different dyes are being used. So quinacrine, okay, it is a technique. One technique is quinacrine, Q banding. Other technique is G banding. Other technique is N banding. Other technique is C banding. So quinacrine banding, it has been proposed by Kasperson in 1958. GMSA binding, it has been proposed by some scientists in 1971. Nuclear organizer region. So NOR region, on chromosome there is one NOR region, that is nuclear organizer region. So the Matsui and Sasaki, they have discovered this technique in 1973, whereas Linde and Lawson, they have discovered this C banding technique, that is centromeric region, where the centromeric region is getting stained. Okay, so that is discovered in the year 1978. So if you look at the Q banding technique, where quinacrine mustard stain is used to stain the chromosomes. So Q banding technique, you can see the output of the Q banding technique. So region rich in AT bases, region rich in GC bases. So there we can find the dark bands, the region which is rich in AT bases, and the region rich in GC bases, that can be indicated by the light staining. Okay, so dark staining, actually it is the ultimate indication of the heterochromatin region, and this light staining, it is ultimate indication of the euchromatin region, because genes are rich in the GC content. Okay, genes are rich in GC content. So the light band which has been occupied by the, that DNA is made up of ATGC, right? So ATGC, like computer knows the language of 0, 1. So the DNA is always ATGC language. Okay, only 4 base pairs are there, 4 nitrogen bases are there, like adenine, thymine, guanine, cytosine. So this quinacrine, the region which is rich in AT bases, so it gives the dark staining, and it is situated in heterochromatin region. Whereas the region rich in GC bases, it is light staining, and it is situated in euchromatin region. Okay, which is gene rich region. Similarly, G banding technique is there. Okay, so it can shows the sulfur rich region, means the DNA which is associated with the sulfur rich proteins. So that can be found out by G banding technique. Okay, G bands visualize the sulfur rich region of the chromosomes, and it also stains the heterochromatin region. Then N banding technique. So N banding technique, it banding pattern is structural non-histone proteins linked to NOR region, that is nuclear organizing region. So N banding bands appear on NOR region. So silver nitrate binds to the NOR region. Okay, and this band represent non-histone proteins which specifically at NOR region. Then there is a C banding technique which gives an idea about heterochromatin, It binds that sodium, it binds to the repetitive DNA. Okay, repetitive DNA means it is present in heterochromatin. Then there is one more technique, that is R banding. So R banding, where GC region will be stained, that is euchromatic region is seen as dark in R banding. Okay, euchromatin region is seen as a dark in case of R banding, that is the GC rich region. I told you the genes are GC rich, and the GC rich region, that is the euchromatin, is seen as a dark when we go for the R banding. So whatever we have seen till now, this is regarding the banding technique. We have seen the shape of the chromosome, how the name of the gene located on the chromosome is given, short arm and long arm of the chromosomes. We have also seen the centromeric position, classification base of chromosome based on their centromere. We have also seen the various banding techniques, which gives an idea about heterochromatin, which gives an idea about euchromatin, which gives an idea about NOR region, which gives an idea about the centromere where it is located. Okay, so there are different kinds of dyes are being used to stain the chromosomes. Now, if you look at the chromosomal aberrations, so in a ideogram, that is the diagrammatic representation of a chromosome, where we can study the mitotic metaphase. The chromosomes are being studied at mitotic metaphase, and where we can find the diagram, where you can see the shape, size and number of chromosomes. If I tell you that in a human being there are 46 chromosomes, so of course at metaphase, mitotic metaphase, you should see the 46 chromosomes. You should see the total 46 chromosomes at mitotic metaphase. But what will happen if there is an only 45? What will happen if you find only 44? What will happen if you find 47? What will happen if you find 48 instead of 46? So that is the chromosome abnormality. That is the chromosomal aberrations. Means you are not finding the whatever you are expecting, whatever the things you should be expecting as a normal. Okay, so there is a change in the variation. There is a change in the number of chromosomes. It may be less, it may be more. So that is the chromosomal aberration. So in case of human being, as I said, there should be 46 chromosomes at the mitotic metaphase. But if you see only the 45, 44, or if you see the more than that 46, like 47, 48, so that is the abnormality. It will produce an abnormal individual. Okay, so that is the chromosomal aberrations. Where so chromosomal aberrations, it can happen by the two ways. One is by structure and one is by number. So it is possible during the separation, during the anaphase, there will be no proper separation of the chromosomes during the anaphase at two different opposite poles. There is no proper separation of the sister chromatids to two different opposite poles. Means it will some at one pole there will be more number of chromosomes, at other pole there will be less number of chromosomes. Okay, so there will be a change in the number of chromosomes, and that will lead to the genetic deformities, that will lead to the genetic disorders, that will lead to the genetic diseases. And there is also possibility that there will be change in the structure of the chromosome. Means there during the separation of chromosome during the anaphase or cell cycle, there it is possible that some part of the chromosome is being deleted, some part of the chromosome is being duplicated, some part of the chromosome is translocated. Okay, so whatever with respect to the number of chromosomes, we call it as a numerical chromosome aberrations, and whatever is possible, it is relevant to the structure of the chromosome, like there will be a deletion of the chromosome, say part of chromosome, there will be duplication of the part of chromosome, there will be translocation of the part of the chromosome. So that is the structural changes in the chromosome, that is structural aberration. So numerical means instead of 46, you can find 45 or you can find 47. That is the numerical chromosomal aberration. So there are two types of chromosomal aberration, like structural chromosome aberration and numerical chromosomal aberration. So when we talk about changes in the chromosome and their symbol, you can see the changes in the chromosome symbol and how they are represented. So here we can see whenever we talk about the monoploid, that is only one copy of a single genome, we said that it is represented by X symbol, small x. Monoploids are represented by the symbol X, that is small x. Haploid, that is the gametic chromosome complement, it is being denoted by the small n. Diploid, two copies of genome, it is denoted by 2x or 2n. The polyploidy means more than two copies of a genome or two copies of each of the two or more genomes. So autopolyploid means genomes are identical with each other. So there are three copies of one genome. So autotriploid. Autotetraploid means four copies of one genome, that is represented by 4x. Autopentaploid, five copies of one genome, 5x. Autohexaploid, six copies of one genome, that is represented by 6x. Autoheptaploid, that is seven copies of one genome. Means copies of a genome. Okay, so like suppose X is the basic chromosome number, we can call it as a monoploid X. So if that X is means 2x, 3x, 4x, how many times that genome? That is because of genome duplication. That can happen during the course of evolution. Okay, autohaploid, seven copies of genome. Allotetraploid, that is the two copies each from the two distinct genome. Okay, there are presence of two distinct genome, two copies from two distinct genome. Allohexaploid, which the wheat is an example of allohexaploid. So two copies each of the three distinct genome. Allooctaploid, two copies of each of the four distinct genome. Okay, and there is the other term that is the aneuploid. Okay, euploidy means we have seen about the changes in the entire genome, means like 3x, 4x, polyploidy. Okay, autopolyploidy, allopolyploidy. Entire genome is getting duplicated. So aneuploid, there will be a condition that is monosome, one chromosome is missing. So as I said you, if I expect the 46 chromosome there, and if I can find only 45, means it is the condition like 2n minus 1, means one chromosome is missing there. And that condition we call it as a monosomic. Double monosomic, one chromosome from each of the two different chromosome pair is missing. So it is denoted by 2n minus 1 minus 1. Nullisomic, if one chromosome pair is missing, if there is a missing of chromosome number 1, pair means it is a homologous chromosome, right? So if the chromosome number 1 pair is missing, means it is 2n minus 2. That is the condition we call it as a nullisome. So these are all chromosome abnormalities. These are all chromosome aberration. Trisomic condition means one chromosome extra. So if I expect the 46 chromosome being there, so there will be 46 chromosome, presence of 47 chromosomes, means one chromosome is extra. So that condition we can call it as a trisomy. So then there are some structural chromosomal aberrations. So the chromosomal aberration means during the cell cycle, if there is some error during the separation of that chromosomes. Okay, so it can lead to it can happen structural chromosome aberrations. It can happen because of deletion, it can happen because of duplication, it can happen because of inversion, and it can happen because of translocation. So these are the types of structural chromosome aberration, that is deletion, duplication, inversion, and translocation. So deletion means some part of the chromosome is getting deleted, means it is an intrachromosomal. Duplication means some part of the chromosome is duplicated, it is also an intrachromosomal. Inversion means some part of the chromosome, that gene sequence is getting inverted on a chromosome, that is also an intrachromosomal. But translocation means exchange of genetic segment between the different chromosomes, that is interchromosomal type of aberration. So these all are deletion, duplication, inversion, translocation. These all are structural chromosomal aberrations. So in this diagram, in the middle, you can see there are 6 genes on a chromosome. Let's assume that there are 6 genes on a chromosome. So which are they? A, capital B, capital C, capital D, capital E, and capital F. So if there is a deletion, of C gene, so you can see that C gene, you can see after deletion you can't find the C gene there. If there is an insertion, so there is an insertion of L gene, you can find out the insertion. So other gene has been inserted there. So that is the type of insertion, type of structural aberration. L gene has been inserted, whereas C gene has been deleted in case of D gene. And native gene sequence is A, B, C, D, E, F. In case of inversion, you can see A, B, C, D. So it is getting EDC. Instead of C, D, E, you can find there EDC, means inverting. So what was the order of gene? It was C, D, E. But right now, what the order you can see there? It is E, D, C. Then duplication. So duplication here, which genes has been duplicated? You can see the D gene has been duplicated. So this is the structural chromosome aberration. This can lead to the errors, the genetic disorders, genetic diseases. Okay, inversion, here you can see the some part of the gene is being inverted. In case of duplication, D gene has been duplicated. In case of deletion, C gene has been deleted. In case of insertion, L gene has been inserted there. So this inversion, deletion, insertion, duplication, these are the intrachromosomal changes, intrachromosomal structural changes or structural aberrations. Whereas translocation means it happens between two non-homologous chromosomes. So there is an Here we can see there is an exchange of genetic material between two non-homologous chromosomes. That's why there is the gene sequence has been changed. Constitution of a chromosome has been changed. So native gene sequence was A, B, C, D, E, F. The homologous chromosome was having the gene sequence as L, M, N, O, P, Q. And when the translocation has happened, so there is an exchange of genes within the non-homologous chromosome. So right now you can see on one chromosome capital A, capital B, capital C, and small OPQ. That OPQ gene has come from its non-homologous chromosome. That means there is an exchange of genetic material. So this translocation is also a type of structural chromosome changes. Okay, structure. Right now we can say about it as structural chromosome aberration. These all of five are the types of structural chromosome aberration. So the deletion. So I told you that in deletion, so A, B, C, D, E, F. So here E and F genes are being deleted. So deletion is associated with some genetic disorders. So like Cry-Du-Chat is there. Okay. So small portion of chromosome number 5 is being deleted. Then Philadelphia is there. Deletion of chromosome 22. Prader-Willi syndrome is there. That is because of deletion of long arm of chromosome 15. So there are different genetic orders which are result of that deletions. Okay. Structural chromosome aberrations. Then duplication. So here we can see A, B, C, D, E, F. It is a native gene sequence, original chromosome. And there you can see A, B, C, D, E, F. And again E, F. So E, F gene has been duplicated. The two genes, the chromosome segment has been duplicated. Okay. So duplication. So duplication, then now inversion. So inversion, right now you can see the A, B, C, D, E, F, G. A, B, C, D, E, F, G. This is the native gene sequence. And you can see there A, B, C, then it comes F, E, D, means some part of that particular gene sequence has been inverted. Okay, it has been inverted. So instead of D, E, F, you can see there F, E, D. There are two types of inversions like pericentric and paracentric. So here we can see in paracentric inversion, the centromere is not involved. So here we can see that horizontal green color line indicates the centromere. Okay. So between E and F gene, there is a centromere. So here we can see A, B, C, D, E. And you can see the B, C, D. The gene sequence has been inverted there. Here you can see the DCB, means gene has been inverted. The orientation of gene has been changed. Okay, right now you can see the gene order on a chromosome A, D, C, B, A. Okay, so this is the paracentric inversion, means here the centromere is not being involved. Then pericentric inversion, where actually centromere is involved. So here in between E and F, there is a centromere. So up to F, there is an inversion of gene sequence. Okay, we call it as a pericentric inversion. So orientation of that gene sequence has been changed. Instead of DEF, right now you can see FED. Okay, that is the result of pericentric inversion. Translocation. So in translocation, I told you that translocation happens between the non-homologous chromosome, means within the chromosome, interchromosome. So whatever other structural chromosome aberrations we have seen like deletion, duplication, inversion, that can happen within a chromosome, intrachromosome. But in translocation can happen within a non-homologous chromosome, interchromosome, other chromosomes. Okay, there is an exchange of genetic segment. So in simple translocation means what you can see there. So in above green color part, red color part, you can see A, B, C, D, E, F, G, H, I, J, K genes are there. Then there is a centromere, and it is followed by short arm, that is P, Q, R, S, T, U. They are the genes which has represented on a non-homologous chromosome with some small alphabets. And what happens after translocation? And there is another non-homologous chromosome which is represented by numbers A 1, 2, 3, 4, 5, 6, 7, 8. So what happened? There is an exchange of genetic segment. So from one side of the chromosome, AB has been deleted, AB genes has been deleted, and that has come on the other chromosome. That is because of translocation. Okay, that AB gene has been deleted from its original position, and that has been translocated on its non-homologous chromosomes. That is the kind of simple translocation. Okay, it is the simple translocation. Then this is the reciprocal translocation. Reciprocal translocation means, so their genes are being exchanged between the two non-homologous chromosomes. So A, B, C, D, E, F, G, that was the gene sequence on one chromosome. H, I, J, K, L, M, that was the gene sequence on its non-homologous chromosome. And after the reciprocal translocation, you can see H, I, J has come on first chromosome, that is blue color chromosome, and AB has come to the orange color chromosome, that is ABKLM. That is a reciprocal translocation. Okay. In case of simple translocation, the segment of gene has been deleted from one position and it has been inserted at the other non-homologous chromosome. Here we can see the AB gene, which has been inserted on the non-homologous chromosome. So that is the, now whatever we have seen, that is the structural chromosome aberrations. Whatever we have seen till now, it is the structural chromosomal aberrations. So now we can look at the numerical chromosome aberrations. So structural chromosome aberrations, what structural chromosome aberrations we have seen? We have seen the deletion, we have seen the duplication, we have seen the inversion, we have seen the translocation. So that was relevant to the structure of the chromosome, either deletion of segment of chromosome, duplication of segment of chromosome, or in translocation of segment of chromosome. But right now we can look at the numerical chromosome aberrations. Numerical chromosome aberrations are those that cause a change in number of chromosome. So number of chromosome is being changed. I told you, if I expect the 46 number of chromosome in each somatic cell of an human beings, then if there is a change, if there is a variation in the number of chromosome, instead of 46, if there will be 45, instead of 46, if there will be 47, one extra chromosome. So that is the related to the number of chromosomes. So that's, we can call it as a numerical chromosome aberrations. So it is being classified into the two types, euploid and uneuploid. So euploid means those whose somatic genome is the exact multiple of the basic number of characters of the species. So we have seen the polyploid, like 1x, 2x, autopolyploid, like triploid, tetraploid, hexapentaploid, hexaploid, octaploid, heptaploid. So this means set of a genome is duplicated. Okay, entire set of a genome is duplicated. Entire set means entire set of a genome is duplicated there. We call that situation as euploid. But in case of uneuploid, those in which somatic number is irregular multiplication of basic number. Occasionally, this could be reduction in the basic chromosome number also. Uneuploid means either one chromosome is lacking, like if I expect the 42 chromosomes in rice, and if I can see only 41, if I can see only 40, if I can see 44 instead of 42, means it's kind of aneuploidy. Okay, it's kind of aneuploidy. Aneuploidy means change in the chromosome number. Okay, and euploidy means change in the entire genome, entire set of n chromosomes. So here we can see numerical chromosome aberrations are divided into the two types, that is euploidy and aneuploidy. So euploidy means entire set of n chromosome is multiplied there. From n there is a diploid, then polyploid, triploid, tetraploid, pentaploid. Entire set of a genome is duplicated in case of euploidy. Whereas in case of aneuploidy, there will be condition like hyperploidy and hypoploidy. Hyperploidy means, what are the examples of hyperploidy? It is a trisomy, tetrasomy. Whereas hypoploidy, it has the example like monosomy and nullisomy. Okay. So we'll look at what are these aneuploidy. Aneuploidy means, I told you that if there is one extra chromosome, like 2 plus 2n, if that 2n is equal to 42, if rice has chromosome number 2n is equal to 42, And instead of 42, if you find the 43, means 2n plus 1 means 42 plus 1. So 43 chromosomes. So that condition is trisomy. If you find 44 instead of 42, so the condition is 2n plus 2, that is tetrasomy. If you find monosomy, if you find 41 instead of 42, then it is 2n minus 1, means 42 minus 1, then 41. The condition we call it as a monosomy. Nullisomy means 2n minus 2, means if you find only 40 chromosomes, so 42 minus 2 means 40. So it is a nullisomy. So there is 2n minus 2, two chromosomes are missing. Monosomy, one chromosome is missing. Trisomy, one additional chromosome is there. Tetrasomy, two additional chromosomes are there. So that is the aneuploidy. Euploidy means entire set of the chromosome is being duplicated. So euploidy has various type. Haploid, you know, the gametes are haploid. So two gametes are fused together. Diploid conditions can be formed. And 2n, from 2n, if we will give the colchicine treatment, it will be polyploid like 3n, 4n, 5n. So this is regarding the numerical chromosome aberrations. And whatever we have seen previously, it was structural chromosome aberration like deletion, duplication, inversion, and translocation. So euploidy means the change in chromosome number which involves the entire set of chromosome. The change in chromosome number which involves the entire set of chromosome. Euploidy have one or more complete genomes which may be identical with or distinct from each other. Because euploidy also have some types like autopolyploidy and allopolyploidy. That's why, which may be identical with or distinct from each other. The somatic chromosome number of euploid individual is exact multiple of basic chromosome number of that species. And what are the examples euploidy includes? It includes monoploids, it includes diploids, it includes polyploids. So entire chromosome set is being duplicated there. And in case of aneuploidy, it is because of some lacking of one chromosome, lacking of two chromosomes, or there will be more two chromosomes extra, there will be more one chromosome extra in a cell. Okay, that conditions we call it as a aneuploidy. That means addition of one or more entire chromosome or loss of such a chromosome. Addition of one or more entire chromosome there or loss of one chromosome. So we are looking at the euploidy now. We will talk about the entire set of chromosome. Okay. So entire chromosome set is duplicated. Right now we are looking at numerical changes, numerical aberrations of the chromosomes. In a numerical aberration, there are two types: euploidy and aneuploidy. Euploidy means entire set of chromosome is getting duplicated there. In case of aneuploidy, I told you that there will be presence of one extra chromosome, there will be presence of two extra chromosome, there will be lacking one chromosome, there will be lacking two chromosome. That is the aneuploidy. So in euploidy, so first is monoploids. So monoploids contain single chromosome set, and which is denoted by the small x letter. Okay, which is denoted by the small x letter. And monoploid differ from haploids. We know that haploids we are representing by the symbol small n, which carry half of the gametic chromosome number. If 2n is equal to 42 means n is equal to 21. If 2n is equal to 46 means n is equal to 23. Means each gamete will have the half of the chromosome. Okay. So haploids, gametes are haploid in nature. They carry half of the chromosome number, or we can call it as a gametic chromosome number. In true diploid species, both monoploid and haploid chromosome numbers are same. Okay, x is equal to n. So what is the difference between monoploids and haploids? The difference between monoploids and haploids. So haploid, you know that monoploids, you can see, represent gametic chromosome number of a diploid species, whereas haploids represent the gametic number of any species. It may be polyploid. So wheat, you know, it is a hexaploid. So if wheat has the chromosome number 42, means its haploid chromosome number will be 21. Okay, monoploids is something different. So monoploids are always haploids. Haploids cannot be always monoploids. So monoploids means basic chromosome number. Like if you see the wheat example, so wheat is an allohexaploid. So actually basic chromosome number of wheat is 7. Okay, 7 is the basic so monoploid number, basic chromosome number of the wheat is 7. So many monoploids are always haploids. It contains single set of chromosome. It may contain one or more copies of genome, because in case of polyploid, haploid is represented by the small n symbol. Then diploid is the characteristic by the presence of two genomes in a somatic cell. Two genome means 2n. We call it as a 2n. Means whenever we say in diploid organism, so we say that 2n means there are pair of homologous chromosomes. So each homologous chromosome is coming from either male parent or female parent. Either it is of maternal origin or it is of paternal origin. Then polyploids, the organism with more than two genomes are called as polyploids. Among plants, polyploid occur in multiple series like triploid, tetraploid, pentaploid, hexaploid, heptaploid, octaploid. So that is extra because of basic chromosome number, and thus resulting in triploid, okay, tetraploid, etc. So generally, ploidy levels higher than the tetraploid are not commonly encountered in the natural populations. However, there are some exceptions. Example: hexaploid wheat, octaploid strawberry. See, 8x, 6x. So what is this small x? It is the monoploid number. It is the basic chromosome number. So if I say to you the basic chromosome number is 7, so 6 into 7 total is equal to 42. So that's why wheat is having 42 chromosomes. So basic chromosome number of wheat is 7. But it is denoted by X symbol here. Okay, monoploid. But 6, if I say you that x small x is equal to 7 in case of wheat, that is basic chromosome number of wheat is 7. So total chromosome will be 6 into 7. That is 42. So wheat will have the 42 chromosomes. And if you have to, if anybody asks you to what is the haploid number of wheat, you will tell that it is half of the 42, means 21. So strawberries, they are the octaploids. So types of polyploid, right now we are looking only at the euploids. We have not touched the aneuploids. So what are types of polyploids? So polyploids are distinguished on the basis of source of chromosome into three main kinds. That is allopolyploids and autopolyploids. So autopolyploids means multiple copies of identical chromosome set, usually developed normally. Allopolyploid means multiple copies of non-identical chromosome set. So allopolyploid, wheat is the main example in allopolyploid. Okay, wheat is the very common example for allopolyploid, where different type of genome has been incorporated in a wheat, different species. That's why it is a allopolyploid. Auto means its own genome is getting duplicated. There is no any horizontal transfer of a gene. Okay, so its own genome has been duplicated in case of autopolyploid. So autopolyploids in a plant, when the same set of chromosomes of a genome are increased in a number, so autopolyploids are obtained. So it involves the homologous chromosome sets. Okay. So in a many species, autopolyploids shows an increased vigor, size, and this phenomenon we can call it as a gigantism. So actually during the course of evolution, many plants you can see they are the polyploid in nature. They have the more genetic constitution as that of native or ancestor parent. During the course of evolution, you can find the polyploids are being more evolved because to adapt to such changing climate conditions. Polyploids are more adaptive because they are having the more genetic content in that, because of duplication of an entire genome, its own genome, and they are also shown the increase in the vigor, size, and this phenomenon we can call it as a gigantism, and it can happen naturally. It can happen naturally. So polyploids means larger cell size than diploid. larger prolonged range, slower growth and late flowering, larger leaves, flowers, fruits. Okay, so these are the various properties of autopolyploids. So autotriploid, there are some examples of autotriploid. Here we can see autotriploid have three complete set of genomes of the same species in somatic cell. Means basic chromosome has number has been duplicated thrice, 3x. So it is autotriploid, 3x. And what are the examples of autotriploid? Triploids are useful in what are the examples like banana, sugarcane, apple, sugar beet. Okay, and if you see, actually triploid do not produce a true seeds. Okay, that is the disadvantage of that polyploid. Then autotetraploid. What are the examples of autotetraploid? Groundnut, potato, coffee, grasses, alfalfa, rye. So in autotetraploid, 4 copies of a genome of same species. Here we can see capital A is one kind of genome and it has been duplicated 4 times. A means one chromosome has, you can find out if there is a chromosome number 1, you can find the chromosome number 1 4 times, because entire set of a genome, entire set of a chromosome has been duplicated 4 times, and that's why it is an autotetraploid. 4 copies of each chromosome, 4 copies of entire set of a chromosome. Okay, so what are the examples? Rye, alfalfa, grasses, groundnut, potato, and coffee. These are the examples for autotetraploids. Now come to the allopolyploids. Whatever we have seen, auto means in euploidy we have seen autopolyploids. In euploidy we have seen diploid, haploid, monoploids, and autopolyploid. So autopolyploid means where entire is segment of the genome has been duplicated. But in case of allopolyploid, there is also a duplication of the genome, but actually it is come from the other species. A polyploid containing genetically different chromosome sets. A polyploid containing genetically different chromosome sets from 2 or more species is known as allopolyploid. What it is? A different chromosome sets from 2 or more species. Whenever we do the crossing and whenever, so there is an, you know that wheat, how wheat has been evolved. So wheat is the very good example for this allopolyploid. It contains the chromosome sets are not homologous. There are some distinct. They are come from the various species during the crossing. Okay. So natural allopolyploids can be produced natural during the course of evolution. So wheat is the most common example for that, because whatever the crossing with that rye and triticale has been produced, so that is because of that natural crossing, interspecific crossing. It happens naturally, and this leads to this has led to the chromosome doubling. Okay, and ultimately the plants were having the multiple set of chromosomes, and that is from the different species, and we call it as a natural allopolyploids. Artificial polyploids can be generated. Okay, so artificial allopolyploids means, so if we do the crossing, it cannot happen naturally. Okay, we have to do the crossing, we have to incorporate some gene from one species, we have to incorporate some gene from either species, and in this process so whatever we have seen now, it is regarding the euploidy, means entire genome has been duplicated there, like monoploid, like monohaploid, diploid, triploid, hexaploid. Then we have seen allopolyploid, autopolyploid. So entire genome has been duplicated there. Okay, entire genome. But now we can see the other type of numerical. What we are seeing now, structural chromosome aberration we have seen. We have also seen the numerical chromosomal aberration. In numerical chromosomal aberrations, we have seen that euploidy and aneuploidy. Euploidy means entire set of an chromosome is being duplicated. Entire means one chromosome will have 3 copies, one chromosome will have 4 copies, 5 copies, pentaploid, like that. And in case of aneuploidy, as I told you, there will be lacking of one or more chromosome, or there will be loss of or addition of one or more chromosome. If the chromosome number of wheat is 42, then if chromosome number of wheat is 42, means that wheat crop or the cell which will have the either it will have the 41 chromosome, means one chromosome will be lacking, or either it will have the one chromosome extra, the trisomy conditions. Similarly, if it is a rice plant where 2n is equal to 24, so there is a possibility that either one chromosome, either one cell will have 23 chromosomes, or other cell will have the 25 chromosomes. Means one chromosome either it will be extra, one chromosome either it will be less. So that is the condition that we can call it as an aneuploidy. So there are various types of aneuploidy. We will see that. So these are the kind of aneuploidy. These are the types of aneuploidy. That is this numerical changes in the chromosome. First is monosomy. If I expect there should be a 24 chromosomes, and if there is an one chromosome is lacking, means normal chromosome number if it is 2n is equal to 24, and if I found the 23 chromosomes there, means one chromosome is lacking. So the term for conditions we can call it as a 2n minus 1, means 24 minus 1, so 23 chromosomes, and the condition it is monosomy. Nullisomy, 2n minus 2, two chromosomes are lacking, means 2n minus 2. If it is a 24 chromosome in rice, then 2n minus 2, 24 minus 2, it is 22 chromosomes I can see there. Trisomy means one extra chromosome. If I expect 2n is equal to 24, and if there are 25 chromosomes, means 2n plus 1, then that condition we can call it as a trisomy. Tetrasomy, so if there are two extra chromosomes, if 2n is equal to 24, and if I find the two extra chromosomes there, then that condition is tetrasomy, that is 2n plus 2 condition. Okay, so you can see the monosomy is represented by the genomic formula, that is 2n minus 1. The diploid organism which lacks one chromosome of a single homologous pair is called as monosomy. So I told you, if there will be, if I expect 24 chromosomes, so one chromosome is lacking, so 24 minus 1, 23 chromosomes will be there. So that is an abnormal situation. Okay, so it is represented by the formula 2n minus 1. Nullisomy, it is represented by the formula 2n minus 2. If the normal chromosome number is 2n is equal to 24, and if I found there 22 chromosomes, means two chromosomes are lacking, so it is represented by the formula 2n minus 2. That is the condition that we can call it as a nullisomy. That means entire pair of homologous chromosome is lacking there. Means chromosome 1 and its homologous chromosome is lacking. Chromosome 2 and its homologous chromosome is lacking. Means two chromosomes are lacking which are homologous with each other. Trisomy, it is represented by the formula 2n plus 1. So there is a presence of one extra chromosome. There is a presence of one extra chromosome. Means if I expect 2n is equal to 24, so there will be 25 chromosome instead of 24. So one chromosome will be extra. Tetrasomy, so if I expect 2n is equal to 24, if I found there 26 chromosomes, means two chromosomes extra. So 2n plus 2, so that conditions we can call it as a tetrasomy. So all are these related to the numerical and structural aberrations of the chromosomes. Now with this I will stop it here, and just I will summarize whatever we have seen today. We have seen the structure of chromatin. Okay, we have seen histone protein. We have seen DNA protein ratio. We have seen DNA protein ratio there. Then we have seen the histone protein types. What are the different histone octamer is there? How much DNA is wrapped around the nucleosome? What are the nucleosomes? What is the basic unit of a chromatin? What is the primary structure of the chromatin? We have seen what is the sister chromatin, what is the S phase, how the DNA is getting synthesized or how DNA is getting duplicated during the S phase, why DNA is getting synthesized or why DNA is getting duplicated during the S phase of a cell cycle, when in which phase cell organelles are getting synthesized. Then we have seen the various karyotyping. What is karyotype? What is ideogram? That is diagrammatic representation of the chromosome. How the chromosome, what is the long arm, how it is being denoted, small p. What is long arm, how it is denoted q. How the gene is being localized, like q, like 8q. I have given the one example on which chromosome it is local, on which chromosome gene is present, at which position gene is present. So that we can come to know how much heterochromatin is there in the genome, in the chromosome, how much euchromatin region is there. So that can be come by the study of euchromatin and heterochromatin. What is euchromatin, what is heterochromatin, that I have explained you. Then what are the different banding technique. So there are some banding technique which gives the dark stain region for the heterochromatin region. There are banding technique which stains the centromeric position, centromeric region. Then there are some banding technique which stains the chromosomes at euchromatin. There are some banding techniques which stains the chromosomes at NOR region. Then we have seen about the structural aberrations of chromosome, deletion, duplication, inversion, translocation. Then we have seen the numerical aberrations of chromosome, that is euploidy or aneuploidy. In euploidy, we have seen the autopolyploidy and allopolyploidy. In aneuploidy, we have seen the monosomy, nullisomy, trisomy, and tetrasomy. So with this, if you have any doubt, you can unmute yourself and you can ask the questions if you have any. Is there any question?